Hi, everybody. In this podcast, we're going to begin to explore different types of cell processes. We've spent the last couple of podcasts talking about cellular structure, and now we're going to get into talking about how cells actually work, now that we have a pretty good foundation of what they're made of. So we're going to jump right in. So in this first part, we're going to look at how cells actually move materials into and out of them. One of the most important things a cell can do is actually move substances around. So that's kind of a big deal. So what's kind of pretty important is that we have this idea of transport. How are we going to get things around? So there's a couple of types of transport that I want you guys to be familiar with. The first two are really important because these are where we're going to look at in this particular podcast. They are called diffusion, and what's ultimately known as is active transport. Okay, so diffusion across the lipid layer, I'm going to get into what that is, but this is super, super important. I mean, like, we're going to spend a lot of time on diffusion. Passive transport is just a way of moving materials around where the cell doesn't actually have to expend any additional energy. It just happens. So diffusion is a form of active transport. Okay, so pretty straightforward. Active transport, just like when you're active, it's, when you're being active, you're using a lot of energy. So we'll start talking about cells and their use of actively transporting materials around, where the cell actually has to use energy to do so. So this is kind of a big deal because the cell actually has to really use additional energy, and in most cases, when cells use energy, this is what they're primarily using it for, is this idea of active transport. And then these last two aren't really as important, but they're big ways of moving materials around. So for endocytosis and exocytosis, this is, endo means I'm going to bring something into the cell, so it's kind of a big way of really engulfing like a huge amount of stuff in. Exocytosis would be the cell like expelling a whole lot of material. So we aren't, it's just there to kind of be, recognizing the fact that cells move things in other ways. We're going to be focusing, though, pretty heavily in the second half of this unit on these first three types. So let's really get into the idea of how things begin to move across a membrane. All right, so here's the deal, okay? We know a cell membrane, in terms of how it's structured, shows selective permeability, okay? So that cell membrane in that lipid bilayer, okay, has selective permeability. This term is a really important term that you get a handle on, okay? All selective permeability means is that some things can cross and others can't. It's pretty much that easy. Some things are able to move freely across that lipid bilayer. Other things need some help. So let's look, okay? This, if I'm talking about this substance here, okay, these substances that are going straight through, those would be the fact that the membrane is permeable to those. If substances can't cross that bilipid layer, then the membrane is impermeable, okay? So it would be impermeable, which just means things can't cross, and these substances would be permeable. And so what I want to do is kind of give you a few examples of what those are. So some substances that the membrane that the membrane's permeable to are things like oxygen, carbon dioxide, other small nonpolar molecules. Don't worry about what nonpolar means. It just means that it has a little bit of a, unequal charge, and some water molecules. So in essence, little molecules can just go right through, okay? Things that are impermeable are big molecules like glucose, proteins, water-soluble molecules, and then ions. Even though ions are really little, okay, ions are atoms that have a charge. That's what an ion is. Okay, and so even though they're little, because they carry a charge, okay, things like this, that means they can't just zip right through that lipid layer. And just as a reminder, that right here, remember our little head and tails? There's your phospholipid molecule. So that whole membrane is made up of a gazillion phospholipids. And remember, they're not bound, they're just there, and you can just go right through. So one of the ways that molecules do pass through across the membrane is through a process known as diffusion. And this is a really important process to understand. We use it a lot. And our cells are constantly having to either allow diffusion to happen or in essence have to fight it. And that's where their energy comes in. So diffusion, this definition is key. This is going to be our working definition. You're really going to have to understand this, and we're going to do a lot of really awesome labs that are going to showcase this. But all diffusion is the net movement, so net overall, right? So overall movement of molecules from an area of high concentration, so where there's a lot of them, to an area of low concentration. Okay, so that overall movement until equilibrium, so that just means evenness, is reached. So for example, if I have a beaker or a bucket or a glass jar full of water, all these little blue doodads are water, and I put some red food coloring in there. Notice here, these are all very, very highly concentrated molecules. So there's my very high concentration. Okay. Now these molecules, because molecules bang, 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 bang around, these molecules are going to begin to spread out, and there's going to be an overall movement of molecules into this direction because over here there is zero red molecules. So that's my low concentration area. Okay, so these molecules are going to continue to spread out until they're evenly spread out, which is all equilibrium is. So a little while later, notice they're starting to spread out a little further, and then after a certain amount of time, they are very evenly spread out. Okay, so I go from being really highly concentrated over here to starting to spread a little bit more. Notice that area is a little bigger, and now they're completely spread out between all of the water molecules, which are your blue dots. And you can even do that at home. Just put a drop of food coloring into some water and just let it go. And if you leave it long enough, you're going to find that it's completely even throughout the water. So that's all diffusion is. That's how it works. Now, there are things that are going to influence how fast diffusion happens. And we are going to do a most neat lab. Well, I think it's neat because it's student designed, which I really like. A few years ago, a student actually designed this lab, and it's called the Diffusion Box. And we are going to do this in class. And what the Diffusion Box does is actually explores some of these things that's going to influence the rate, right, speed, at which diffusion happens. So some of those factors could be the steepness of the concentration gradient. Oh my, concentration gradient, there's a whole nother term. Okay, gradient, really important that we know that. Okay, all gradient means is a difference on one side of the area to the other. If you go back to what I just showed you with the food coloring, at the initial place of dropping in that water, there was a gradient. There was a lot in one area and a little bit. I like to think of gradients, are just like a hill. Okay, so when I think of concentration, here's where I have a lot of what I have. Okay, and here is the little bit of what I have. If there is a difference, let's say I separate that area. So now I have a high area over here. I have little to none over here. That's a gradient. Now how steep that is, so if I have a really steep gradient, okay, where there's a whole lot on one side, okay, that's going to influence how fast diffusion happens. It's going to happen faster where it's steeper. Think of rolling something downhill. The steeper the hill, the faster it rolls, right? It's the same idea. So steeper gradient, faster diffusion. Pretty straightforward. Other factors that influence how fast diffusion happens is going to be the size of the molecule. Okay, we're going to be talking about big molecules and little molecules, which can be tough to understand given the fact that we're still talking about something we can't see with our eye. These are molecules. But molecules have size. Some are very tiny, some are really big. And the bigger the molecule, the slower diffusion is going to be. In fact, if it's too big, diffusion won't even happen at all because it won't be able to cross the membrane. So, smaller molecules diffuse faster. Temperature. Okay, temperature is really going to influence how fast diffusion happens because some of you guys might already know this, but as I add heat, I'm adding energy, and that causes molecules to move really fast, right? So the faster they're moving, the more likely they're going to spread out a lot quicker because that's what drives diffusion in the first place, are those little molecules bing banging around. So there you go. Higher the temperature, faster the diffusion. If I lower temperature, things really start slowing down and diffusion slows. And lastly, other types of gradients like electrical gradients or pressure gradients are going to influence how fast diffusion happens, okay? So in our cases, though, we're going to be spending really most of our time really checking out these. But I just wanted you to be aware that these also, you know, the idea of electrical or pressure gradients also influences the rate at which diffusion happens. So to kind of wrap up today, we're going to talk about one form of diffusion that is so important we give it its own name, and that is called osmosis. And you guys may have heard about the term osmosis, but this is a really, really important concept for living things. Our cells being mostly made of water, our environment for our cells is all water, so that movement of water is really key. Here's your definition of osmosis. Boom, done. What? Okay. The diffusion of water. That's it. So if I know what diffusion means, which is the movement of molecules from a high concentration to low concentration, in this case I'm not just talking about anything, I'm talking about specifically the movement of where water is highly concentrated to where water has low concentration. And that is just has its own term called osmosis. It's diffusion, it's still passive, the cell doesn't have to put in any extra energy to make it happen, just does. So let me show you a little idea of what I mean about this, and we'll explore this as well. So this is great because this is just giving you a little insight into things to come. So if I take a bag that has 2% sucrose, okay, now here's the kicker. Bag, okay, I'm going to give you guys some terms here. The bag is permeable. The bag is permeable to water. It is impermeable to sucrose, which is just table sugar. That's all it is. All right, so I want you to think back in your head, what does that mean? Okay, so permeable versus impermeable. The bag is permeable to water, impermeable to sucrose. So if I take this bag and I put it into a situation where it's in distilled water, that means outside the bag, right here, my concentration, I have 100% water. And I want you guys to think, in the bag, if I have 2% sucrose, how much does that leave? You know, what's my percentage of water? Got it? Write it down. Were you right? Yeah, you guys are smart. Okay, so 98% water. So when I put this bag in that begins with 98% water, in here, where is my higher water concentration when I start? Is it inside the bag or outside? Well, I think you're all aware that 100 is more than 98. So that means that there's more water outside the bag than there is inside. So water is going to diffuse in, causing my bag to swell. Now, if you notice, there is this tiny little arrow going out. But wait, why would water leave? Here's the thing. Individual molecular motion is random. Just because molecules are moving, remember that diffusion, that osmosis, is an overall movement. Some little molecules are still going to sneak out. They're going to exchange. They're just bouncing around completely randomly. So that individual molecular motion is random, okay? Random. So some will go out, but the overall, notice the size of the arrow is much bigger coming in. My bag is going to swell. All right, so that's beaker one. Let's take that same bag, 98%, and let's put it into a solution where I have 10%. So if I have 10% sucrose, you guys can do the math. I have 90% water. So I put it in, my 98 started in here. That is more water inside the bag than out. Water is going to osmose out of the bag, and my bag is going to go and shrink way down, okay? Because there is more water in than out, water leaves. The overall motion of the water is going to be out. So now, in beaker 3, I'm going to take a 2% sucrose solution and put it in a solution of 2% sucrose, which leaves me with 98% here. Well, guess what? They are even Steven, right? Totally equal. That means my bag is already at equilibrium. And because it's already at equilibrium, the motion of the molecules, there's still going to be exchange. Water is still moving because it can, but there's not going to be any overall. There's going to be no net change of water as it crosses. And that's how it works. And I can actually see this if I use blood cells. So here I have a red blood cell right there. Okay. Now these are scanning electron micrographs of blood cells that are put into solutions that have different concentrations compared to what's inside. So I'm taking my red blood cell and I'm putting it in. Well, first, let's go over here because this is your equilibrium. So here's what the red blood cell normally looks like. See how it has that sort of donutty indent. So that's what a red blood cell should look like. Now, if I put it into a solution where there's more water outside than in, notice how swollen these cells are over here. Super swollen, right? Because water's flowing in. In this one, there was more water in the cell. Look what happened to my cell. It went shrivel, 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 right up. So even cell, so this shows us that water can move across that cell membrane without the cell having to do any work. So this is something that we can actually witness, which is pretty cool. So with that, we're going to end today because in the next podcast, we will be looking at the process of active transport, which is going to be bringing energy into the system and to see what happens. And we'll explore that as well when we do the Diffusion Box lab. So that's all. So just look, we'll do a lot of things to explore this idea of diffusion, but that's really it. So we will see you in class and have a great night. Talk to you guys later.